Hello students, greetings for the day. Hope you all are doing well and learning with great zeal and enthusiasm. The topic of today's video is determination of blood cell count. So in today's video, we will focus on how the blood cells are counted in the laboratories. Alright, so in today's video, we will be covering that how many blood cells are there in the human blood. We will focus on the determination of these blood cell count, that how with using different methods the different blood cells are counted in the laboratory. So a complete blood cell count is usually performed in the hematology laboratory for the determination of blood cell count of the human blood. So it is one of the most common laboratory tests ordered by the clinicians even for a routine health checkup of a healthy person. CBC or complete blood cell count is ordered to ensure there is no underlying disease when the individual may be asymptomatic. Alright, so without delaying, let's start the video. So what is a blood cell count? We need to understand this at the very first thing, that what is actually a blood cell count. It is a measure of the number of red blood cells, white blood cells and platelets in the blood. So as we all are aware, we have also discussed the different types of blood cells in the blood. We have discussed the composition of blood in the previous video, which is named blood. So in that video we discussed that blood consists of three kind of cells, and these are your RBCs, WBCs and the platelets. Here you can also see that the blood cells are RBCs, white blood cells and the platelets. So the complete blood cell count or the blood cell count is the measurement of the number of red blood cells, white blood cells and the platelets present in the blood. Alright, the amount of hemoglobin which is present in the RBC is also considered under the complete blood cell count. Alright, and the hematocrit, that is the packed cell volume or the amount of whole blood that is made up of the RBCs, the amount of hemoglobin concentration average present in each RBC. These kind of measures also comes under the CBC test. So there are different parameters which come under the CBC test. Number one is the red blood cell count. Number 2 is the white blood cell count. Number 3 is the platelet count. Then there is hemoglobin concentration, and there is hematocrit, which is which consists of the red cell indices such as MCV, MCH, MCHC, and the RDW, which is called as red cell distribution width. It also consists of hematocrit, which is your PCV, packed cell volume. So these all parameters come under the CBC test. One more parameter is there, which is called as DLC, which is the differential leukocyte count, the different types of leukocyte count. Alright, so we will focus on all of these counts. In today's video, we will focus on the method through which we count red blood cells, the method through which we count white blood cells and platelets in the blood. Alright, so without delaying, let's start the video. So there are three type of blood cell, as I told you. These are RBCs, WBCs and the platelets. The blood cells are formed in the bone marrow. They begin their life as stem cells and then they mature into these three types of cells, that are RBCs, WBCs and the platelet. So CBC consists of numbers that are printed out from the hematology analyzer. So CBC is performed by the hematology analyzer, as well as it can be performed by the manual methods also. So various parameters are there, as I told you. RBC count is there, hemoglobin level is there, hematocrit is there, RDW, MCV, MCH, MCHC, reticulocyte count, the WBC count, the DLC, differential leukocyte count, the platelet count, mean platelet volume, and the platelet distribution or differential width is also there in the CBC count. So it is all about the CBC, that what parameters are constituted in the complete blood cell count. Now a type of blood cell that is made in the bone marrow and found in the blood. What is RBC? Now first of all we will discuss about the RBC count, that how RBC count is performed in the laboratory. So what is RBC? RBC is called as red blood cell. It is formed in the bone marrow by the erythropoietic precursor cell, which are formed in the stem cell. Then they mature and develop and into different stages. They undergo differentiation into different stages, and then they mature into red blood cells. Alright, so red blood cells consist of a protein which is called as hemoglobin. We all know that hemoglobin is a chromoprotein. It is a colored protein which carries oxygen from the lungs to the all parts of the body. Now what is the principle of this red blood cells count? Alright, so red blood cell can be counted by using an automated hematology analyzer, which is also called as autoanalyzer, and it can also be counted manually in the lab. There is let me check, let me see here. So it can be counted by using this manual chamber, which is called as improved neobar chamber. Manually in the lab we charge this sample with the blood sample, diluted blood sample in the reagent, and then the blood cell are counted by using microscope. So this is also the method through which the blood cell can be counted. Alright, so let's focus on its principle now, that how this red blood cell count is usually performed in the laboratory. So RBC count is a blood test that the doctor prescribe to you to know how many red blood cells count is there in your blood. It is also known as the total erythrocyte counting, because RBCs or red blood cells are also known as the erythrocyte. Why this test is important? It is important because RBCs contain hemoglobin, which carries oxygen to your body's tissue. The number of RBCs you have can affect how much oxygen your tissues receive. Your tissue needs oxygen to function, to perform different vital functions that are required for the proper functioning of our body. Alright, so total erythrocyte count can be performed in the laboratory by three methods. First one is the photoelectric counting method. Second one is the electronic counting method, and the third one is the hemocytometer counting method. So the first two methods are performed by the autoanalyzers, which are present in the laboratory, through which what we do, we just have to mix the sample of blood into an anticoagulant vial, and then we have to aspirate that sample of blood, the EDTA sample, into the electronic autoanalyzer. The electronic autoanalyzer will aspirate the amount it needs for performing the total erythrocyte counting, and then after few minutes it will give you the printed result. Alright, so in today's video we are going to focus on the manual method, that how manual method is performed in the laboratory, because using autoanalyzer is not very much difficult, but to know about its principle is very important, that how the red blood cell are counted, what is the mechanism that is gonna happen through which we are counting the RBCs in the blood. It is important. So thus in this video we are gonna discuss about the manual method through which the red blood cells are counted in your blood. Alright, so is a special type of chamber is used for counting the blood cells in the specimen, alright, which is your EDTA blood. The special chamber is called as the hemocytometer or the Neubauer's chamber. You can also see here, it is written here that it is called as the hemocytometer or the Neubauer's chamber. So for this blood, the method we are using here is the Hayem's method. Alright, Hayem's method is so called because of the reagent we are using, Hayem's fluid. It was given by the scientist Hayem's. So what we do, we mix the EDTA blood. We collect the specimen of EDTA blood from the patient. We collect the blood into EDTA vial, then it is called as EDTA blood. We mix the EDTA blood with the Hayem's fluid. Alright. This is present commercially in the laboratory. Alright. Commercially prepared Hayem's fluid is there. So we mix the EDTA blood with Hayem's fluid in the ratio of 1:200. Alright. So why we are using Hayem's fluid is here for the counting of red blood cell is this, that the Hayem's fluid is isotonic to the red blood cells. What does the isotonic word means? Iso meaning same and tonic or tone means concentration. So we can say that Hayem's fluid has the same concentration as the concentration of RBC. Thus, Hayem's fluid is used for the determination of the count of the RBCs. Alright. So Hayem's fluid does not causes any damage to the RBCs. The normal saline solution is also used, but by using it, there is slight creation of the red blood cells, and Rolex formation can also be seen in the RBC. Rolex formation is the aggregation of RBC on one another. This can be seen. If the Rolex formation occurs, the RBCs cannot be counted properly. Alright. Thus, Hayem's fluid is generally used for the determination of red blood cells. Alright. So after diluting the specimen, the content is charged on the hemocytometer or the Neubauer's chamber, and then there is a specific area in which the red blood cells are counted. The red blood cells are counted in the middle area or the center area of the Neubauer's chamber. Alright. Let's see the Neubauer's chamber or its details in the next slide. So here for using the hemocytometer chamber or the Neubauer's counting method, we have a hemocytometer chamber, cover slip, light microscope, RBC pipette and the RBC diluting fluid. Alright. So RBC pipette is the pipette which is used for diluting the sample of blood with the diluting fluid, which is Hayem's fluid. Alright, it consists of the glass pipette which is then attached with the rubber tube, and the mouthpiece is there, but mouth suctioning is not into use nowadays. So what we do, we just push this pipette so that it sucks the amount of fluid or the blood specimen we require for the lab. So what we do, we suck 0.5 up to 0.5 mark we suck the blood sample, and then up to 101 mark we aspirate the Hayem's fluid into this pipette. Then what we do, we mix this blood sample with the diluting fluid and leave it here for 1 to 2 minutes so that the proper dilution of blood with the diluting fluid can take place. Thereafter, what we do, we apply a cover slip on this Neubauer's chamber. This is the improved Neubauer's chamber. It consists of this, when we observe it in the microscope, this type of blocks can be seen. So we count the blood cells in these different chambers. Alright. So what we do, we put a cover slip here, and then we charge the sample that we put into the pipette here into this Neubauer's chamber through using the RBC pipette. After that, the Neubauer's chamber is left for 2 to 3 minutes for a complete charging of the fluid. Then the red blood cells are counted in this mid chamber. Alright, in this mid chamber the red blood cells are counted. It consists of 5 into 5 boxes, that is 25 boxes. 1, 2, 3, 4, 5 and 1, 2, 3, 4, 5. So it consists of 25 chambers, sub-chambers in which the red blood cells are counted. Now, blood should be carefully drawn to the 0.5 mark of the RBC pipette. Then it should be mixed with the normal saline or the HAM solution up to the 101 mark, so that the proper dilution of blood can be occurred. So 1:200 ratio is needed for the red cell counting. Blood and diluting fluid are mixed by shaking the pipette in a horizontal position for 2 to 3 minutes to ensure complete hemolysis of the WBCs. Alright, now 2 to 4 drops of mixed fluid are discarded, and then the tip of the pipette is touched to the side of the hemocytometer chamber, and drop of fluid should be charged by using the red blood cell pipette. Then wait for around 2 to 3 minutes, as this is the time which is required for the settling of the erythrocytes in the Neubauer's chamber. Alright, after this the counting method is performed. Total number of cells in 5 squares in the center of counting chamber is determined under the high power objective of the microscope. Then whatever observation we have to put it in the formula which is here. The number of RBCs or the count of RBCs equal to the number of cells in 5 squares into dilution number into reciprocal of the volume. Alright, so we will put the values we have observed into this formula, and thereby we will get the total amount of erythrocytes in our blood. Alright, so what is the normal RBC range in humans? In males, the normal RBC range is 4.7 to 6.1 million cells per cubic mm or per microliter, and in females it is 4.2 to 5.4 million cells per mm cube or per microliter. Now, why erythrocyte count is performed? It is very important that what is the clinical significance of this erythrocyte count. So, if your RBC count is increased than the normal range is this. In the for example, let's take an example that in laboratory we performed total erythrocyte count, and the result we get for the male person is 6.3. Then we can say that the RBC count of this person is increased, is not as per the normal range, it is increased. So, whenever there is an increase in the total erythrocyte count above than the normal range, then it is called as erythrocytosis. Whenever there is increased count of erythrocyte in blood, it is called as erythrocytosis. It is a clinical condition. So, erythrocytosis can be observed in number of conditions. Alright, it can be observed in burns, cholera, heart disease. It can also be observed in the polycythemia. It can be observed in if the person is having shortness of breath. It can be observed in the tenderness in the palms of the hand or soles of the feet. Alright, it can also be observed if there is a disturbance in the sleep of the person. Now, talking about the decreased erythrocyte count. If there is decreased erythrocyte count than the normal range, then the person is said to be erythrocytopenic, and the clinical condition is called as erythrocytopenia. So, erythrocytopenia can be observed in old age. It can be observed in the case of pregnancy. It can also be observed in the case of Anemia. Anemia is a very common example in which the erythrocyte count goes below the normal range. So it can be observed in the case of dizziness, weakness, or if the person is suffering from anemia. So anemia is a clinical condition in which the concentration of hemoglobin is decreased, the RBC count is decreased, following which the oxygen carrying capacity of the blood is also decreased. So decrease in erythrocyte count can be a possible reason for anemia. Now talking about the white blood cell count. So white blood cell count is also called as the total leukocyte count, or in laboratories we generally call it as a TLC test. The test is often included in the CBC, but the doctor may prescribe you it separately also as TLC. Whenever there is a case of infection or underlying disease, doctor prescribe the test TLC, that get your TLC performed and show us the results, so that they can prescribe you different medications for the test as applicable. So what is the principle of this test? So the sample of blood is diluted with Tux fluid. Tux fluid is the fluid which is called as the WBC diluting fluid, which is required for performing the WBC counting test. Alright, so in the same manner, Neubauer's counting chamber is used for TLC count also, but the diluting fluid is different. Diluting fluid here is the Tux fluid, and the instead of RBC pipette, WBC pipette is used for counting the TLC, total leukocyte count. So what is the difference between WBC pipette and RBC pipette? So the difference between the WBC pipette and the RBC pipette is this, that the WBC pipette consists of white bead and the dilution varies in the WBCs and RBC pipette. In the WBC pipette there is a dilution of 1:20, whereas in RBC pipette there is dilution of 1:200. In the WBC pipette, the lowest mark is 0.5 and the highest mark is 11. Whereas in the RBC pipette, the lowest mark is the 0.5 and the highest mark is the 101. Alright, which then impacts the dilution of the fluid. Alright, so what happens here? The blood sample is mixed with the Tux fluid. The Tux fluid consists of 1% glacial acetic acid and or 1% HCl. So what does this WBC diluting fluid do? WBC diluting fluid does not causes any damage to the WBCs, but it damages the RBCs. It ruptures the RBC completely. The glacial acetic acid which is present in the Tux fluid ruptures the RBCs present in the blood sample and does not cause any damage to the WBCs. Thus the WBCs are preserved, and then we can see the WBCs properly in the counting chamber by using a microscope. The blood and diluting fluid are mixed by shaking the pipette vigorously in a horizontal position for 2 to 3 minutes, so that complete hemolysis of RBCs can take place. Then the first 2-3 drops of the fluid is discarded, and then the Neubauer's chamber is charged using this diluted fluid. Then after charging the Neubauer chamber, it is again kept for 2 to 3 minutes to let the leukocytes settle down in the chamber. Now for RBCs, we counted the sample in this mid chamber, but for the TLC count, for the total leukocyte count, the leukocytes are counted in these 4 corner chambers in a zigzag motion like this. Alright, first the chambers also differ. In the mid center there is 5x5 box, that is 25 boxes are present, but at the corners the number of sub boxes are 4x4, that is 16 chambers are present at these corners. Alright, So total number of cells in 4 squares at the corner of this counting chamber is determined under the low power objective and high power objective of the microscope. Then the observation we get from by performing the total leukocyte count is then put into the formula in this formula. The total number of WBC is equal to the number of cells in 4 squares multiplied by dilution number into reciprocal of chamber depth. Alright, so by putting the observation in this formula, we will get the result of the total WBC in our body. So what is the normal range now? So the normal WBC range of in the person of the blood is 4,000 to 11,000 WBCs per microliter, alright, or 4,000 to 11,000 WBC per mm cube. Alright, now why the WBC count is performed? So the condition in which the WBC count is increased above the normal range is called as leukocytosis. So if the WBC count increases above then 11,000, that it may be considered as leukocytosis. There may be different clinical condition or the disorders of WBCs or the diseases in which the leukocytosis can be observed. So leukocytosis can be observed in leukemia, which is the cancer of WBC. In this the WBC count is increased manyfold the normal range. Then leukocytosis may also be observed in the cases of myeloproliferative diseases, in which there is a disorder in the proliferation of the myeloid series of the WBCs. Then there may be, in case of infection also, the WBCs are increased. So to rule out any kind of infection or inflammation in your body, TLC can be prescribed by your doctor. Then the clinical condition in which your leukocytes, the WBCs, goes down or decreases than the normal range is called as leukopenia. So leukopenia can be observed in number of diseases or clinical conditions such as typhoid. In the case of typhoid, the WBC count may be lowered. In case of anemia, dengue, influenza, malaria and the aplastic anemia, leukocytes are decreased. The leukocytes can also be decreased in the case of immunocompromised diseases or the immunosuppression diseases such as TB or AIDS. Now the platelet count. So platelet count is the another number of blood cell count performed in the patient's blood. Platelets are also called as thrombocytes. So the platelet counting test can also be called as the total thrombocyte count or the total platelet count. Alright, so why platelet count is performed? Platelet count is performed to see if the person is normally having platelets or is suffering from any of the disorder or the disease in which the platelet count is decreased. Due to the decreased count of platelet, if there is some clinical condition that has occurred in the patient. So in the same way, platelet count is also performed using the Neubauer's chamber. So what happens in the in this also a different diluting fluid is used. So diluting fluid used here is the ammonium oxalate fluid. It is mixed in the ratio of 1:200. All right, 2 micro this 0.2 ml blood is mixed with 380 ml dilution fluid, and then the Neubauer's chamber is charged, and then after charging the Neubauer's chamber, the cells are counted in the mid chamber of the Neubauer's chamber. Alright. So what we have to do, we have to prepare the ammonium oxalate by mixing it from a still standpoint, because this reagent has a tendency to settle down at the bottom of the flask. Then we can also use it to dilute blood So it will destroy, what is the principle of the test, that it will destroy the RBCs and the WBCs, and the platelets are preserved in the sample. Then we will mix the blood sample with the diluting fluid and leave it for 2 to 3 minutes. Then we will charge the Neubauer's chamber by using this diluted fluid and leave the chamber so that the platelets can be settled in the Neubauer's chamber. Then we will place the chamber inside a petri dish with wet cotton and cover it up with a lid and wait for 15 to 20 minutes. Then the cells are counted in the middle chamber, which consists of 5 into 5 boxes, and then it should be put in the formula for platelets. Alright. So what is the clinical significance of platelets? Why platelet count is performed in the person? So platelet count is also performed because to rule out any bleeding disorder or any disorder of the coagulation also. So the rise or fall of platelet cells indicates several conditions, especially the ones related with the clotting disorder. Alright. It may also be ordered when a bleeding disorder manifests, such as prolonged bleeding, epistaxis, that is the nose bleeding, GI bleeding, or the heavy menstrual bleeding. So when the platelet counts are decreased below the normal range, it is called as thrombocytopenia. It can be observed in the case of anemia or ITP, which is called as idiopathic thrombocytopenic purpura. Alright, which is a result of antibody attack against the platelets. It can be also observed in the case of HIV. The platelet count is decreased in the case of dengue. Also in the case of typhoid also the platelet count can be observed that it has decreased. Then in the case of aplastic anemia, the platelet counts are decreased. Now whenever the platelet counts are increased above the normal range, it is called as thrombocytosis. So it may be the result of malignancy, any kind of malignancy or cancer causing condition. Then in the case of TB also the platelet count may be increased. In the case of rheumatoid arthritis, the platelet count is observed to be increased. Then whenever a person is taking any birth control medications, it may impact the count of platelets of the person. So that is all about the determination of blood cell count in the laboratory. We discussed about the 3 types of blood cell and by which methods these 3 cells are counted in the laboratory using different diluting fluids or reagents, and what is the principle of mechanism of these tests. Hope this video helps you. Keep learning, keep studying. Thank you.